In this video, we're going to talk about the angiosperm life cycle. And like all plants, angiosperms exhibit the alternation of generations. As mentioned, the angiosperm life cycle includes an alternation of generations. And like all plants except for the non-vascular seedless plants, the sporophyte generation is the dominant generation. And by dominant, we just mean that it is the most noticeable. The sporophyte generation, remember, is multicellular, and the cells are diploid. And diploid just means that the cells have two sets of chromosomes. One set of chromosomes comes from the maternal gamete, which is the egg, and one set comes from the paternal gamete, which is the sperm. Angiosperms are heterosporous, which means that they have separate male and female structures involved in sexual reproduction. So let's start by doing a quick review of the female structures that we talked about in the other video when we talked about flowers. So if you recall, this entire structure up here is the gynoecium, which is also known as the carpel. And the gynoecium is further broken down into more specific parts. So the top of the carpel is known as the stigma. The neck-like part of the carpel is the style. And then this area down here is known as the ovary. And within the ovary, we have these ovules. An ovule in the surrounding tissues is known as a megasporangium. So megasporangia is plural. Megasporangium would be singular. So the ovule and some surrounding tissues make up the megasporangia. Within these ovules, there are megasporocytes, and these cells are diploid. And these cells are ultimately going to go through meiosis in order to form haploid megaspores. And some of these megaspores are going to develop into the female gamete, which is the egg. So the megasporocytes are located in these ovules here. And then you can see on the diagram that they are going through this process of meiosis to form 4 haploid megaspores. You can see those 4 megaspores in this picture here. Out of these 4 megaspores that develop, only one of them is actually going to survive and move on to the next stage of development. And I accidentally put this picture on the wrong slide, so you can ignore it for now and we will talk about it on the next slide. The megaspore that survives at the end of meiosis is going to go through mitosis 3 times. So if we draw that down here, we've got our megaspore with our haploid nucleus. After 1 round of mitosis, we're going to have 2 nuclei. After another round of mitosis, we're going to have 4 nuclei. And after a third round, we are going to have 8 nuclei. Now, usually with mitosis, cytokinesis also occurs, and that is the case here, except for these 8 nuclei at the end of the 3 rounds of mitosis are only going to be in 7 cells. So one cell is going to have 2 haploid nuclei. And you can actually see those cells in this picture here. So if we take a look at one of these ovules here, and that's what this is showing here as well. I'm going to kind of draw it on its own out here. So if this is one of those ovules, within that ovule, there are going to be 7 cells. On one end of the cell, there's going to be an egg cell with a nucleus and 2 synergids, which are kind of smaller helper cells. At the other end of the ovule, you're going to have antipodal cells, and you're going to have 3 of those, each with their own nucleus. And then in the middle of the ovule, you're going to have 1 cell, and that center cell is going to have 2 nuclei in it. You can also see those cells on this diagram here, where you can see the 3 cells on one end of the ovule, 3 cells on the other end of the ovule, and then that center cell with 2 nuclei present. Now we'll do a quick review of the male structures associated with angiosperms. So if you recall, this entire structure up here is known as the androecium, also referred to as a stamen. The head of the structure is known as the anther. The stalk holding it up is known as the filament. And inside the anther, you have the pollen grains. And technically, they're not pollen grains until they get to this point down here, but we'll talk about the sequence of events that gets us to that point. The pollen sac, also known as the anther sac, is the microsporangium. And it is located in the anther of the androecium. Within the microsporangia, there are sporocytes, and sporocytes are just a type of cell. And in this case, these are diploid cells, so they have those 2 sets of chromosomes. And these 2N sporocyte cells are going to go through meiosis. And remember that during meiosis, we reduce the ploidy of the cells. So a 2N cell ends up producing 1N daughter cells. And so the 2N male sporocyte goes through meiosis to form 4 haploid 1N microspores. These microspores are then going to go through mitosis, so the haploid cells are going to produce more haploid cells, and they are ultimately going to form a pollen grain, which is the male gametophyte. We can see that happening in this picture over here. So we start out with the microsporocyte, which is diploid. And it goes through the process of meiosis to form 4 haploid microspores. So you can see the 4 microspores kind of connected to each other here. So we have 1, 2, 3, 4. So then we'll take a look at what happens with just one of these microspores going through the next stages of development. So each of those microspores is going to go through mitosis, which you can see here, and it's also going to develop a tough pollen coat around it. So this spiky bit here, that is the pollen coat. And because mitosis happened at this arrow here, we have two cells contained within this pollen grain. So this is one pollen grain here. And it has two cells located in it. And each one of these cells has a haploid nucleus. So to recap that, if we take a look at this microspore here, this microspore has a haploid nucleus in it. And it is going to go through mitosis to form two cells, each with their own haploid nucleus in it. Now these two cells are going to be surrounded by a pollen coat which helps to protect the cells inside. Now these two cells are going to have different functions associated with them. One of these cells is going to be a pollen tube cell. And one of them is going to be a generative cell. That is ultimately going to go through mitosis again to form two sperm cells. The pollen is released from the anther when the anther dehisces. And dehisces is just kind of a fancy word for breaking apart. And so basically the anther is going to dry out and split apart, and the pollen is going to be released from inside of it. Now let's take a look at what happens during pollination. And remember that pollination is not the same thing as fertilization. In plants, pollination has to occur before fertilization can occur. During pollination, a pollen grain lands on the sticky stigma of the carpel. Now remember that inside this pollen grain, we have three cells. We have a tube cell and then two sperm cells. The tube cell from the pollen grain is going to start forming a tube or a passageway through the style, which is this neck region here. And so that pollen tube is going to start moving down through that style to eventually deliver the sperm to the cells in the ovule. And you can see the zoomed in kind of picture of the ovule over here. And you can see that pollen tube forming and coming down and eventually entering the ovule through this opening called a micropyle. And then those sperm cells get deposited into the embryo sac, which is what this structure is now referred to as. Recall that in the embryo sac, there are 7 cells present. There are 3 antipodal cells, a central cell, an egg cell, and then 2 synergids. And each of these cells has a haploid nucleus except for the center cell, which actually has two haploid nuclei present. Remember that the pollen grain had one tube cell and two sperm cells present. So we actually end up with something called double fertilization. One of the sperms from the pollen grain is going to fertilize the central cell. And remember that the central cell has two haploid nuclei in it, and the sperm cell also has a haploid nucleus. So when that sperm fertilizes that central cell, what we end up with is a triploid cell. And triploid just means that it now has three sets of chromosomes in it. Two sets of chromosomes came from the female cell that was produced, and one came from the sperm. This triploid cell is then going to go through mitosis in order to form something called the endosperm. This endosperm is going to end up nourishing the developing embryo inside the seed. The cells of the endosperm are going to be able to produce carbohydrates, proteins, and lipids, and these can be used by the embryo to make energy in the form of ATP, and also to form other compounds and molecules that it needs as it grows and develops. Recall that from the previous slide, one of the sperm cells in the pollen grain fused with the central cell to develop into the endosperm. The second sperm cell from the pollen cell is going to fuse with the egg cell also in that embryo sac. And when the haploid sperm fuses with the haploid egg, it forms a diploid zygote. And a zygote is just a unicellular structure that now has a diploid nucleus in it. The zygote is then going to go through mitosis in order to form an embryo. So the zygote goes through embryonic development in order to basically make more cells until it develops into a new organism altogether. Within the developing embryo, there is a radicle, which is a small root that forms, and then one or two cotyledons. And we'll talk about what a cotyledon is more when we talk about the difference between monocots and dicots. But a cotyledon is a passageway for nutrients from the endosperm and the seed to the embryo. And you can see the cotyledon here. That's what this kind of whole structure is here. And then a seed coat will also form, and the seed coat just serves a protective function to protect the embryo developing inside. So to review the structures of a seed, we have that seed coat, which is the toughened outer layer, and its primary role is protection. And the word integument just means a covering. As humans, we have skin, and our skin is actually known as our integument, and it's part of the integumentary system. The seed also has that endosperm, which has the kind of food reserves to help to nourish the developing embryo. And then the embryo itself is the developing baby plant. So to come full circle with the life cycle of an angiosperm, remember we're talking about this alternation of generations. So if the seed lands in a suitable environment, it is going to germinate. And germinate just means that it's going to go through mitosis over and over again to ultimately form a new multicellular diploid sporophyte. So the plant, the kind of flower itself, is the sporophyte. And then remember that in the center area of the plant here, we have the carpels, also known as the gynoecium. which contains the female gametophyte. And then we also have the male gametophyte in the androecium, which were the stamen, and they contain the male gametophyte. So you can see how the cycle continues because the gametophytes within the sporophyte plant can then produce more sperm and egg, and sexual reproduction can happen again to produce a seed that produces another sporophyte that produces gametophytes, etc., etc.